Good afternoon. I will start to present this work by saying that, as we know, after infection, HIV disseminates and establishes in different compartments of our body, where it can cause a latent infection among these cells despite antiretroviral therapy. Actually, ART is able to stop viral replication and to bring plasma viremia to undetectable levels, but it is not able to eradicate the so-called HIV latent reservoir. That is mainly composed by cells that harbors defective proviruses that are not able to replicate. A smaller fraction of these cells are able to produce viral mRNA, and an even smaller subset is able to produce viral protein. But the most clinically important reservoir is the so-called replication-competent reservoir that harbor intact, that is composed by cells that harbors intact proviruses that are able to reinitiate viral replication in case of ART is stopped or if it fails for any reason. Here we can see the different pattern of decay of two important reservoir markers that are cell-associated DNA, or CAD, and cell-associated RNA, or CAR. CAD represents the total amount of HIV integrated DNA, and we can see from the graphs that it drops quite a lot during the first year of ours, but then it reaches a slow decaying plateau that current therapies cannot eradicate yet. On the other hand, CAR reflects in some way the residual viral replication in ART-treated individuals. It drops by about 1-2 logs, but less with respect to the drops that we observe in cell-free RNA. Here we can see regarding HIV DNA, the different decay of defective proviruses and intact proviruses in treated individuals under viral suppression. Defective proviruses account for more than the 90% of all HIV-1 DNA integrated proviruses, and they are structurally damaged, so they cannot replicate. On the other hand, intact proviruses represent only less than the 10% of proviruses, but they are replication competent, and we see that they have a different decay kinetic with respect to defective proviruses because they drop a bit faster during the first years of antiretroviral therapy by about the 16% per year. So in this context, our study tried to answer a key clinical question, and the question is if switching from a 3-drug regimen to a 2-drug regimen can affect the latent reservoir of HIV. Actually, some non-inferiority trials, such as the SALSA, the TANGO, the PASO-TOBLE trials, and others, have demonstrated that this switch is able to maintain viremia suppression, but less is known at the moment on the effect of this switch on the latent reservoir markers. So in this context, we investigated the reservoir dynamics of these 3 markers that are cell-associated DNA, cell-associated RNA, and intact proviruses at enrollment, T0, and after 18 months, T1, in 38 people living with HIV on stable 3-drug regimen. 18 maintained this regimen, while 16 switched to a simplified 2-drug regimen. For this study, blood samples were processed to obtain and isolate PBMCs. 10 CD40 cells were purified using an automated magnetic separator, and nucleic acids were extracted using a manual guanidine thiosanate-based method to guarantee optimal yield and minimal fragmentation. Then RNA and DNA were analyzed with digital PCR using a QIAcuity machine. CAR was assessed with a simplex D PCR targeting the 5' LTR of the HIV genome, while CAD and IP were assessed targeting the 5' LTR for CAD and the Psi and the M region for IP. Sorry. So here we have the first slide of the results of our study, and we can see the baseline characteristic of the overall population and of the two different groups. We see that no baseline significant differences were observed, and so the two groups were comparable at baseline. And this is important to be confident with the fact that if we see some differences in the results, they can be attributed to treatment intervention rather than to baseline imbalances. Here we can see that comparing T0 and T1 among in all the population and in the two treatment groups, we didn't observe any significant changes in the three reservoir markers, likely suggesting that regimen simplification in this 18-month period does not affect these three reservoir markers. After that, we wanted to understand if we could detect some correlation at baseline among the viral markers that we analyzed and some key clinical parameters, but we didn't find any significant correlation. On the other hand, we found a quite strong correlation among cell-associated DNA and cell-associated RNA at baseline, and this may suggest that the fraction of integrated DNA may reflect in some way the residual viral replication of the virus. Then we wanted to understand if we could find some predictors of the viral reservoir markers over time among the baseline parameters that we had available. And about the variation of CAD over time, we didn't find any strong association, but we found a trend for larger delta CAD when there was a higher level of T0 CD4 and a decreasing T0 cell-associated DNA. About delta IP, we found two quite strong factors that may predict a lower delta IP over the observation period, that are higher viral load and higher baseline interprovirus fraction. While we didn't find any parameters that could predict variation of cell-associated RNA over time. So these are quite few samples, but currently we are performing the same analysis on additional samples. We have enrolled about 18 people, but for the moment we can say that the size of the viral reservoir measured in terms of cell-associated DNA, RNA, and IP remains stable in the medium term of the 18 months of our study in virologically suppressed patients, independently of regimen simplification. Actually, the switch to a two-drug regimen seems not to affect in a measurable way the three markers that we analyzed. Moreover, at baseline, CAD and CAR were correlated with each other, but not with the intact provirus fraction, and this may support a different clinical role of this marker. Also because while the variation of CAD and CAR over time was not associated with any of the key parameters that are usually taken into account in the clinic, IP variation seems to be correlated and predicted by zenith viral load and the intact provirus fraction at T0. So obviously, we can conclude saying that a longer follow-up is needed to understand if the measurement of these viral markers, in particular of the intact provirus fraction, could be of clinical benefit before treatment simplification. And I thank my group of the University of Siena, the group of Rome Tor Vergata that provided us with the samples, and every one of you, thank you.